toothpaste, shaving foam, is it solid, is it liquid? These are mysterious things, okay? So, and then on the top of that, when your shaving foam becomes active, how do you deal with that? It better not. So a lot of this is discussed in this beautiful review by Sriram and others in Hydrodynamics of Soft Active Matter, Reviews of Modern Physics in 2013. Yes. Active solid means it can, I'm not thinking, in solid you don't think of, say, it can change shape, absolutely.
Basically from the response. Yeah, so for instance, proteins are considered solid and they can change that kind of.
So not proteins, but collection of proteins like this cytoskeleton will be considered solid, but it is also active solid in the sense that if you look at its fluctuation dissipation relation, you will see that there is disobeying of fluctuation dissipation. So that's precisely what I will come to. Okay. So this will probably make it clearer. Okay. So absolutely. So this is the phenomena that in cell biology they call flickering. So what they do is here there is a cell and they're using essentially the principle of Newton rings to find out what is the fluctuation of height in this cell. So there are some people sitting right over there, Tanmoy and their gang, who do all these things. And this is a picture that is taken of their experimental setup. So this is called interference reflection microscopy. If you do that, you will, for example, this is how a cell will show up. Okay. So this is what is called a HeLa cell. I believe it's a family of cancer cells coming from someone called Henrietta Lacks. And so if you, for example, what Tanmoy and people do is that they can actually find out what is happening over this very small green square. And they have something like 12 cross 12 or 20 cross 20 resolution in that. And these colors are denoting the height. So different time you get different patterns of color. So that essentially how the membrane is fluctuating in both space and time. Okay. So this is called flickering. You look at the flickering data and now you want to interpret the flickering data. To interpret the flickering data as a physicist, you will start by writing down a Hamiltonian or some energy functional, right? So what is the fundamental quantity in the flickering data? It's the height above something. So you say then the energy should depend on the height. And the total energy, of course, will be an integral over the whole plane. The first term, grad h square, is a surface tension term. So this is the membrane where there is a surface tension. And the second term, del square h whole square, is the curvature of the membrane. So this kappa is a curvature modulus. So this is the model you start with. And then you say, okay, so this is the energy and this is in thermal equilibrium. So the partition function is e to the minus beta h. From the partition function, I can calculate all correlation because I know Gibbs statistical mechanics, right? Everyone here knows statistical mechanics. Well, I can ask their teachers. Okay, fair enough. So what else being done here is, I'm looking at the height fluctuation, which is a function of space and time, and then I am Fourier transforming it. So x is now becoming wave number q, and the spectrum of height fluctuation has a particularly simple expression like this. Okay, hq, h of minus q, has kBt divided, this sigma is actually this gamma. Forgive my notational problem. This, see, this del square becomes the q square. This del to the 4 becomes q to the 4. Okay. And you get this expression. So you take this data that you get from Tanmoy, and then you Fourier transform it, and then you try to fit this. You know the temperature. At small q, you get the surface tension. At large q, you get the curvature. Okay. So that is how you find out one way to find out elastic modulus from the membrane. And you would say, okay, now I know what is the bending modulus of a cell membrane, and I know what is the surface tension of a cell membrane. Okay, except this would be wrong. This would be wrong because we have used equilibrium statistical mechanics, and as I am trying to convince you, this system is not equilibrium. This is not in equilibrium with its environment because it's a living cell, right? So there is a mistake being done here, and the whole thing is trying to understand that mistake. Okay, but now you see, one of the first places that I saw this mentioned is way back in 1951. So it's not that we are saying something very novel here. Even the first people who started doing this kind of things, flicker phenomena in human erythrocyte. Erythrocytes are what are called red blood cells. So typical, this is for HeLa cell, but people have been studying it for red blood cells quite a lot. And back in 1951, people realized, and they knew that this is not equilibrium, but this was the first approximation they did. And now the question is, how do we go beyond that? Now, going beyond that are two different ways of looking at it, and I will do them one after another. So one of the papers, and some of you are familiar with this paper here. So this is a paper that was in Nature Physics in 2016. So what they're doing is they have this red blood cell, and they have attached 4 beads to it. Yes. They're stationary, but they are not equilibrium. Equilibrium always means equilibrium with a heat bath. So they are stationary, but they are not equilibrium, not thermal equilibrium. So this is the red blood cell, and you attach 4 beads to it. 3 of them are held, and this one is like a probe bead. Okay. So what you do is, on one hand, you deprive this RBC of ATP. Okay. So ATP, adenosine triphosphate, is the engine of the cell. So if you deprive it of ATP, it cannot do its active process anymore. But I guess everyone knew this. Okay. So you deprive this of ATP. At the same time, you put a sinusoidal force in this bead. Okay. So you put a force, which is the blue one, and you look at the response, which is the green one. From this, you can calculate a response function, and it is the imaginary part of the response function that is being plotted here. Okay. Response as a function of time, the same bead. Okay. So you basically, you're putting a force and see how is this bead responding, and it is not going with the force. There is a lag between them, and that quantity essentially is being plotted here as a function of frequency. Okay. And then you can do a theory of this, and this is essentially the theory that you will do if you do in equilibrium with fluctuation dissipation relation. And you see that they match reasonably well. This is after this RBC has been incubated in a place that deprives it of ATP for 48 hours. But if you don't do that, then you get these 2 different curves. One is the case where you have deprived it of ATP, which is this one, and the other one is the top one, which is the normal response of an RBC. This difference is the presence of activity. This difference is what is telling you that this is not equilibrium. And this is one of the first papers which is quantifying which part of the fluctuation is not equilibrium and which part is. So you can see that in a high frequency modes, the fluctuations are in equilibrium. In small frequencies, they are not. At high, I don't know. So it's also possible that high frequency, you don't detect it so well. So exactly this, so you can try to do a theory of this. And I will show you a little bit later what kind of theories they do, but those theories are limited. Those theories are limited by what I call the Anna Karenina principle, and I will come back to that. Yes. So see, the dead RBC essentially is an ATP depleted RBC. So that's exactly this response. So this is the ATP depleted one, these square ones, and this top one is the one which is the normal RBC. So that's exactly what they are doing, comparing the dead and alive, but by dead, they mean the ATP depleted RBC. I think Tonmoy have these things that they give some sugar but it basically cheats the cell and doesn't make it develop RBC. I'm sorry, develop ATP. You mean these frequencies? Yeah, this is about 1 hertz. This is 10 hertz, absolutely, yes. So you are saying what biological process in the cell is associated with that? I can't really tell you. Oh, that could be. That could also be. So this experiment has not been done for all kinds of cells. It's possible, most likely. So at the moment, I know only the RBC result. So you can try to, I'll come back to that. You can try to associate this with physical process, right? What happens in the RBC at these time scales and so on. That is an ongoing work. People are doing that. Rungit, no. It is, I would say that over the whole experiment, it is in a stationary state. But it is not in equilibrium. No, I would think not, because over the time scale you are measuring, there are at least 2 things that are happening in it that are not equilibrium. One is that the membrane typically has ion pumps on it. And to stay alive, it opens some, closes some, absorbs something, throws something away all the time. So absolutely right, but that's the test here. Anyhow, this is not my work, but the points are perfectly valid. So that is the test here. Here is a theory of the equilibrium case, which is not difficult to do by calculating the response function, and they are plotting on top of it their experimental data. Which one do you mean here? Lower frequencies. This one. Yes. You mean here? You see, you're basically seeing small frequencies means large time, right? So this is basically saying larger and larger time you look, far and far it is from equilibrium. So it's kind of agrees with your intuition, right? If you are actually looking at time scales this small, then you don't have much difference from equilibrium response. See, here it's not that you can control which process is there and which process is not. So that, I believe, is a very difficult thing to control. But that's what people who, I believe, try to kind of say, look, what will happen if I stop certain process? Will there be something where this point will come and agree with equilibrium? Then I would say the response there is dominated by that kind of process. Okay.
Okay, can I ask a question? Maybe a bit elementary.
Yes, please.
So you'd say that it is not in thermal equilibrium. That means other than half kT, some other energy is involved in the system. Can it be thought in that way?
I would not. I would think that the essential, maybe you can, but I would like to think that the essential property of non-equilibrium is not energy, but entropy. That's the point I will try to make.
Okay. So energy itself
for
is
them, yes. For them, yes.
Not that important.
Rajesh, can you please say that again? I was distracted.
No, that means that energy itself is not that important.
I would like to argue that, but that's not necessary. I mean, that's not, I would like to argue that.
So, but in the case when the frequencies are larger, so it basically follows the some
It
kind of
becomes close to equilibrium. See, it may
equilibrium.
also be that where the frequencies are larger, the differences are smaller, and you will need even more accurate experiment to notice them.
Okay.
Right? Your response of the experiment also becomes worse at higher frequencies. So that's also, there is a measurement bias in the whole problem also.
Okay.
Right.
Yeah.
You
Okay.
had a question. No, behind you. But that's okay. So you are saying that when it is ATP depleted, the cell will shrink.
Yes.
It's not dead. It is just ATP depleted.
Yes, but you also mentioned that somebody asks that is this is this cell dead? Then you mentioned
That was Rongit, and he was always trapping me.
okay, yeah, okay.
So I think for them, what they're saying is that the ATP depleted version is the non-active version. It's possible that the ATP depletion, the shell would shrink a bit. But I wouldn't know much about that. Maybe I will ask Tanmay. Okay. Okay, he hasn't seen any shrinking.
No,
and he's right.
That is in RBC, so it happens in RBC.
Okay, fair enough.
Yeah, no, is it that simple that you can say remove ATP depletion is fairly easy process, I think. So does it imply that when you simply suppressing ATP would lead to equilibrium statistics?
No, but they are confirming that. See, after ATP depletion for 48 hours, they are comparing the response function with what they would calculate as an equilibrium response function. And the agreement is not very bad.
Okay.
Sumana.
Yeah, I had a question.
Yes.
So when you are putting these 4 beats, it is to an individual cell, right?
Yes.
So all the measurements that you're doing is for this entire cell.
For this entire cell, absolutely.
So then are we not assuming that the entire cell has whatever the measurements that you're doing, whereas the membrane is actually extremely heterogeneous?
Absolutely
So
right.
that can lead to several issues in both the measurements as well as the results that you're getting from it. Because
you are absolutely right. So the theory that they do is not for the membrane.
okay.
The theory that they do is for a bead attached to a membrane and so on. And it's of course, they have a model of the membrane and so on. So this is all model dependent with certain parameters, but they are not simply taking the data from a membrane simulation.
Okay.
Their theory also has the presence of the beads into the problem.
Okay.
And you are absolutely right. And I think that's also another aspect to what Nilanjana was asking. See, this is a response of the bead, not of the whole cell. So the response of the bead may saturate at high Qs, high frequencies. But yes, but so by that time, I would have hoped to have covered half of my talk, whereas I haven't been able to say a word about my results. But that's good.
So essentially, if you take the data for granted, then what you're claiming is that this experiment by this group shows that system is an equilibrium for higher frequencies.
But can you see, you can't really define equilibrium frequency by frequency.
So, let me finish that question. My concern, not concern, is correct. Normally, if I see a system where I see that a system is in equilibrium at a certain time scale, that would imply the process of communication between that particular system and its bath, whatever is forcing it, is occurring at a time scale which is lower or comparable to that frequency. Now, if that is the case, which is happening at high frequency, for inactive matter, I would expect that equilibrium will hold for lower frequencies of forcing as well, because that's the definition of equilibrium, right? If a communication or collisions in physically speaking, but here the time scale is lower, then your measurement time scale, then it should always exist in equilibrium.
See the time scale.
But here I see a difference. Is that because it's an active matter?
You mean this part?
Yeah.
No. I think the time scale that you are thinking of exchange of individual collisions is much faster than these time scales. So that is way over there.
So you're not even sampling that. Is that what
No,
you're saying?
no way.
Ah,
Because those will be in extremely small frequencies, extremely high frequencies. Yes.
Here you're replacing that with the
Also related to Dipanju's point. If you have a resonance system, non-dissipative, you do its frequency response, you get a delta function.
Absolutely.
If you have a dissipative system, membrane, in this case, the bead is connected. Think about the membrane to be an elastic membrane. If it is perfectly elastic, no dissipation, you would again get that single frequency response. But it has many dissipative components, so you get a flat response.
Also, in
So
this
linear
one,
response, if you do the frequency at very high amplitude, then of course this whole model breaks down. That we are not looking at, they are not looking at.
no.
And
Also,
it is connected to Golam's question also. Typically these frequencies, you expect that a resonant structure probably would be at 10 hertz or so. So this is why you sweep,
see,
or you could have done with a single pulse and then taking the Fourier transform
in
of
this model, you actually don't even have the inertia term.
right.
So this is completely dissipative.
Yeah,
Yes,
I don't
yes, fine.
So the resonance
I
doesn't
don't
arise.
think there is a resonance happening, right? The
Giving an
resonance cannot
example,
happen.
this is spectral density actually.
Wait,
This is a
let-
spectral density.
I will come back to that. Let's see what Rajesh was saying. Rajesh, can you say it again?
No, resonance cannot happen, right? There is no mass. This is a purely,
I would have thought so too.
yeah, purely
Indeed.
kind of what you call
Yes.
mass.
So, okay. So given that the old people were discussing things, the young people understood what we are talking about, right? Okay. Good. Okay, good. See, these days it is fashionable not to put equation on slides. But this is a wonderful audience. This audience understands equations. So there are going to be lots of equations. So now this is what I was alluding to all the time. The clue to non-equilibrium, I would claim, is the entropy generation. So stay with me and it will be fine. These are not very difficult equations. Okay. If they were difficult, I would not be able to do them. So think of a system with some degrees of freedom, which is x, and it's a large dimension, so x mu, and I'm writing an equation for that. You see, I have a first order equation in time, not a second order equation. That is what alluding to the fact we were just discussing before, that this is done in the approximation of zero mass. So the inertia has actually dropped off the equations. So these are not Newton's equations, these are Aristotle's equations. And then there is a force, and the force, and then there is a random force. Okay. And this random force is multiplied by some other function of x. So this is the most general thing you can write depending on how general f and g are. Okay. So these are the xi. Xi is the random force. Okay. So this is what you will call a generalized Lagrange problem. Okay. So you can capture what I have been doing within this framework with a complicated enough f and a complicated enough g. Okay. And you could spend a lot of time doing math of this. That's a lot of fun, but we are not allowed fun at the moment. But this is a stochastic equation, right? So you know how to solve differential equations, some of them. But what does it mean solving a stochastic differential equation? See, it basically means your equation has noise, right? So you solve it the first time, you have some realization of noise, you get a solution. You solve it the second time, you get a different solution. And you solve it again, you get another solution. Xi is the stochastic term. It's random, white in time. It's completely white in time. Okay. If it is not white in time, I would not be able to do the math. Okay, so coming back, what does it mean by solving a stochastic equation? Every time you solve it, you get a different solution, right? So it looks like it's completely useless solving it. But then you have to ask, it is a function of time, but it's an extremely irregular function of time. Because it is white in time, it is different at every different time. So I didn't put an explicit time there. Okay, so that xi is white noise basically. So now you see what you need to ask for is, what do you mean by solving this? Solving this means instead of asking what x is, you ask what is the probability distribution of x? How does that evolve? Given some initial probability distribution, how does it change with time? The equation that gives you the answer is called the Fokker-Planck equation. So to every large equation, there is a Fokker-Planck and vice versa. So every Fokker-Planck equation can be written in this way. You say there is a rate of change of probability, and in the right hand side, there is divergence of some flux. Okay. So this is the probability flux, which you can write in this manner. The first term is typical, the first term is typically called drift, and the second term is called diffusion. The diffusion is kind of the product of G averaged over. There are various stuff, but what you can do is from this, you can calculate the entropy generation rate. And the entropy generation rate is a product of this force, which is this force, this script F, multiplied by the current J. Equilibrium is defined by the flux being 0. Okay. The definition of equilibrium from the Fokker-Planck is that this flux is 0. So if it is 0, your entropy generation rate is also 0. So this is the one that is showing you in a generalized Lajva problem, the kind of problem we are doing. If you are in equilibrium, your entropy generation rate is 0. If you are not in equilibrium, it's something as messy as this. Now, how to estimate this from data? Yes. What is a tensor? This one? Right, because this is a vector. I mean, this is an n-dimensional vector, so that must be, you can think of it as a matrix. So it's a n by n matrix. Most of the time you don't put that noise because multiplicative noise is a mess. But anyway, you can deal with that. It's the n-dimensional space and a matrix, and you can deal with that. In principle, it can get very messy to calculate. So, but the question now is how to generate the entropy, how to calculate the entropy generation rate from the data. See, you get the data, no one told you the model, right? Yes. This one. So you might ask, how does it relate to the equilibrium entropy, right? So if I was doing everything properly, then this would really be the Boltzmann entropy. It will reduce to that. So this is not the entropy, this is actually the entropy generation rate. It's the DDT of the entropy. That I believe you see that from Fokker-Planck formalism. Right, in the you can do the usual Fokker-Planck formalism and go to equilibrium, and you can see that. I often see that with a much more simpler problem. This is too messy.
So in the case of thermal equilibrium, the D will be connected to the diffusion coefficient.
In the case of equilibrium, your J will be 0, right? Because this is the probability flux. The main point of equilibrium is that the probability flux is 0. So that means your sigma with the entropy generation rate will also be 0. But your entropy will be actually maximized.
Yeah, but diffusion constant would come through D?
It will. All viscosity, absolutely. All transport coefficients will come through D. But you see, all transport coefficients are actually non-equilibrium properties, right? Or slightly out of equilibrium properties. Okay.
Yeah.
So now the question is how to get it from data. And I'm really out of time. Okay. So there is this thing invented, which is almost magic, called short time inference scheme, invented by Srikant and Shupriya and someone else, I don't know, but anyhow. So there is a paper in 2020 where they say inferring entropy production from short experiments. I am not going to prove it for you because that will be another talk. But the main result is the entropy generation rate is a maximum over J of this quantity. Okay. So let me walk you through it of how to operationally use this. So I remember I told you about the x. So you get a time series of x mu, mu denotes the vector, and 0, 1, 2 to n, that's the time step. Okay, you get them, and at the same time, in the space of x that you are dealing with, you construct a function d, any function. So it is useful if you construct the function with some basis and with some weights. Okay, let it be any function, whatever convenience thing for you. Okay, take the function and then calculate this quantity, which is the flux. It's the flux in the x space from one time step to another. You calculate the flux, and then from every time step, you will get a value of the flux. You calculate their mean and square that. You calculate their variance, divide it by delta t, and you get sigma. You get an expression, and the idea is you have to maximize the expression with a set of Wm that you are going to choose. Okay, so you choose a set of Wm, get the sigma, then change the Wms a little bit, see if the sigma is getting better, I mean getting larger and larger. That way, in the Wm space, which is a very large dimensional space, you perform an optimization to calculate sigma. Once you have done the optimization and your numerical algorithm has converged, the theorem tells you that this maximum value is the entropy generation rate that you seek. Okay, not completely true. I will qualify that a little bit later, but that's the essence of the algorithm. See, look at the beauty of it. You don't need to know anything about the system. You have degrees of freedom. You calculate this, maximize the problem, maximize it numerically, and any idiot can maximize things numerically these days, like me. And then you calculate this and find the entropy generation rate. Okay. It works for, it assumes that the time series comes out of a Lajva problem. What happens if it doesn't, I don't know. Okay, but yes, that's an important thing. So one of the first places where this was experimentally tested was in Oyon's lab. So then they were doing this simple model, which I call stochastically shaken parabolic trap, and Vishwajit can correct me on that. Sliding parabola, okay? For him it is sliding, for me it's shaken. Anyhow, so the idea is, see, it's very simple. You have a particle in a trap, and then what is the equation of the particle? It's in a trap which is harmonic. So x dot is minus kx. See, there is no acceleration term, that's gone away. Minus kx divided by gamma, and then there is noise. And then lambda is the center of the trap. And now you are stochastically moving the center of the trap, and the center of the trap itself follows an Ornstein-Uhlenbeck process. Okay, this is the simplest system that you can construct where you can test this thing. And the two variables are x and lambda. So here you are plotting x and lambda, and then with different parameters theta, which is basically the amplitude of this noise and the amplitude of this noise. Okay, so for different values of theta, you get different values of lambda and x. So these are the phase trajectories of your system. With these trajectories, you calculate the flux, then you maximize it over functions, you do all of that, and you get a sigma. Okay, and this is the optimization process. This is the optimization process as you are taking more and more steps in the numerical algorithm. After some point, you see that the sigma actually converges. And for this system, you can actually do a theory, a real theory. Okay, so that is the theoretical value. So there is experimental data with the numerical methods agreeing completely with the theoretical value, described all in this paper. So all of that gives us the confidence that this is a perfectly valid method to apply to real data. Okay. So next set of real data comes from Tanmoy. So there is this interference reflection microscopy that I was talking about. From that you get these snapshots. These snapshots, there is a variation in space and a variation in time. But this is a very large dimensional data. This is not x and lambda. And you understand optimization over a very large dimensional space becomes an even more difficult problem, right? So there need to be some kind of dimensional reduction. We tried various things. The thing that worked is something like this. So you take all this data and you calculate the covariance matrix. Then you calculate the first two eigenvalues of the covariance matrix and project all the data along those two eigendirections. So this is what is called the principal component analysis for matrices that are symmetric. And the covariance matrix is a symmetric matrix. If it is not a symmetric matrix, PCA means something else, but let's not worry about that. Okay. So this is after the dimensional reduction. And now these are the two principal components, I1 and I2. And this is what a typical trajectory looks like. This time for the function D, I take a linear combination of the i's and then perform exactly the same optimization, and we get a map of activity. Okay. You see this, and you see a map of activity at different times. Okay. So that's the end result. Right. So the spatial, so I'll just here. Okay. So these are two typical examples. So here you can see the actual cell in IRM. And over here, we are now depleting the cell of the ATP. So first, this is the normal cell. I call it normal, but Tanmay insists on calling it control. So here I am plotting log of sigma. And you can see clearly that this is a very different value from the background. Okay. And then we are depleting it of ATP. And you see the colors are getting weaker and weaker and weaker. So the resolution by which we can calculate the activity here is something like 1.4 cross 1.4 micrometer patch. And one such patch generates something like 10 to the minus 3 Boltzmann constant kb per second for this normal cell. Okay. So this is the lower bound of entropy production rate that we obtain, and I will spend a little bit of time telling you why it's a lower bound. So I hope I tried to convince you that the entropy expression that I told you from the maximization problem is exact, right? I mean, this is proved by real good mathematicians. But what if you don't access to all degrees of freedom, like you do in the present case, right? If you think of the cell, there are lots and lots degrees of freedom and your resolution is limited. So what when you don't have access to all degrees of freedom? Then the entropy generation rate that you calculate is a lower bound. And in our case, what we are doing is, among all the resolution, we are actually projecting along the principal components. So we are still throwing away a lot of stuff. So at the end, this is a minimum, this is the lower bound of the entropy generation rate that one can find out. So this is the cell, for example. You stay out of the nucleus and you break it up in small squares. And you calculate this for every squares, and you plot a probability distribution of the entropy generation rate. The probability distribution looks something like this. As the ATP is depleted, the probability distribution moves. Okay. So you divide the probability distribution by its mean, then take the log and plot it, and all of them collapse with each other. So essentially the mean is changing, but as the ATP is being depleted, the nature of the probability distribution function itself is not changing. Now, some of you may ask me, then how do you calculate this probability distribution function from theory? Okay, the answer to that is there is an ongoing effort. Okay, so this is the main result of this part of the work. There is unprecedented spatial temporal resolution in calorimetry. Entropy has never been calculated up to this resolution ever. So we are very proud of it. Okay, so now I am going to change gear and I am out of time. So should I stop or? Okay, fine. I am doing, see, each square like this has something like 12 cross 12. So yes, so basically for each blue square, I have something like 144 time series, which is a very large dimensional data, and I have to do a minimization in that space, which will become an extremely difficult problem. So that's why I reduce the data by going to PCA, and I basically have just a two-dimensional space. No, the whole series. I take the whole covariance matrix and then project along that. Right. Each of these are those blue patches. This squares is 12 cross 12 or sometime 20 cross 20. Okay, good. So now I am going to change and talk about mechanics of elastic material. That's too noisy. Then I can't see. Okay, the other thing, that's an excellent question to ask. For a flux, to calculate a flux, you cannot do it in one variable, right? If you are in one dimension, there is no flux. So you need to have at least two variables for it to make a loop. And essentially the area of that loop is a kind of a measure of your flux. Okay, so we could. It just becomes more noisy. We could. I think the PCA, you can now say why two principal components? Why not just one? Absolutely, yes. Okay, so now I'm going to change gear and talk about mechanics of elastic material in 3D. This sounds like the most utterly boring subject one could think of, right? If you had an engineering training, some point someone must have taught you something called properties of material, and you must have been sleeping in that class. And if you are a physicist, you may have also had a properties of material class, but maybe you were bunking that class. That's what happens. So, but I'm trying to convince you that this is both complicated and interesting. Okay. So what is easy and what is difficult? So for this, there is always this thing that goes back to Landau, who says that I was born too late, all easy problems were solved, and all beautiful women were married. So easy means linear problems, and not easy means nonlinear problems. Whereas elasticity typically is a linear problem. Okay. So the main quantity of elasticity is a displacement. So you have some elastic material and you are deforming it. This is elastic material and you are deforming it. So you can say every point is going to another point. Okay. So that is this displacement u. And you take a gradient of that and you symmetrize that, you get the strain. Okay. So then you say the stress will be some elastic coefficient times strain. So this tells you that this elastic modulus is a fourth rank tensor. Okay. That should make you complicated enough. Now you would say, look, it is a fourth rank tensor, but I am now going to talk about a homogeneous material. Okay. I am not going to talk about a crystal. If it is homogeneous material, then this fourth rank tensor should be an isotropic tensor. And a fourth rank isotropic tensor can be written in this form with only two coefficients, except there is a catch. It should be both an isotropic and parity invariant tensor. If it is not parity invariant, there will be three. And then that will give rise to a whole new set of elastic materials, which are very cute, but not part of our topic now. They will be chiral elastic material. So anyhow, there are two elastic coefficients. And from this, you can construct an energy. Now that you have an energy, you minimize it, right? You minimize it with respect to the displacement, you get a differential equation. So this is done in Feynman or Landau-Lifshitz. These two elastic coefficients are called two Lame coefficient. And this is how they are related to other elastic coefficients that you know. For example, this is the bulk modulus, this is the Young's modulus, and this sigma is the Poisson ratio. Okay. So any two of them does the job. Okay. So that's the background. Now, from 3D to membranes. Okay. So far, I have been talking about a linear problem, right? Why linear problem? Because the energy is quadratic in U, right? So the forces will be linear. Now, if you want to do a membrane now, you think that I have a 3D material and I make it thinner and thinner and thinner, and I go into a membrane. Okay. So thickness of the material is H, and all these equations are true when H is very small. Okay. Then you can say I will have two kinds of strain. One is the in-plane strain, which is the V, and one is the out-of-plane displacement, which is the W. Okay. And then you can write a strain like that, and then again a same thing as an elastic modulus. And then you can say the elastic modulus is again isotropic. You can get an energy, you minimize it, you get an equation. This is where the cuteness happens. This is a strongly nonlinear equation. This nonlinearity is coming because something like this, you see, can have a very large deformation globally, whereas the deformation is very small locally. Okay, so a small dimensional object embedded in larger dimension gives you geometric nonlinearity. The material itself is not elastically nonlinear. It's the geometry that makes the problem nonlinear. This is called the Foppel-von Karman equation, one of the nastiest equations seen in physics. Fourth order, highly nonlinear, invented by Foppel and von Karman. Foppel was one of the pioneers of elasticity and fluid dynamics in Germany. The other thing he is known for is the father-in-law of Ludwig Prandtl. So that's same Karman. Okay. Okay. So now this is fun. Okay. Any field theorists here today? No? What do you do to seduce field theorists? Good. I have something for you. So now you want to do statistical mechanics of shells and want to look at buckling. Okay. This may look quite nasty, but stay with me. Okay. So you see, previous step, I told you about an energy function, right? That had a fourth order derivative kind of with a b, which is the bending modulus, and some terms here, right? You remember in the membrane, we had a bending modulus and the fourth order term, which became Q4, right? So we are staying pretty close. So now if you do it for a shell, the problem becomes complicated. Earlier, you were thinking about height field above a plane. For a shell, you need to think about height field from a sphere. Your base is a sphere. That makes the problem complicated. So first you cheat. First thing you cheat is you say, I don't want to worry about the whole sphere. I will only worry about a small part of the sphere. Okay. So you can say that small part is like a rising part from a flat surface. And then there are fluctuations from that rising part. Okay. So there is an overall function, z as a function of x1, x2, and then there is height fluctuations on top of that. Okay. If you do that and do various long algebra, this becomes your energy functional, G0 and G1, where F is the displacement perpendicular to the shell. Okay. Just trust me on that part. So now just look at this one. So this one is F squared. This one is F squared. This one is F squared. So this is a harmonic or linear part of the energy. This one is F whole thing squared, so F to the 4. And this one is F and another F, so F cube. Okay. So they are both part of the energy, but this is nonlinear or anharmonic, and this is linear or harmonic, right? Now you want to do statistical mechanics of this system. So as you know that doing the harmonic part is easy, but first you want to non-dimensionalize this. So if you want to non-dimensionalize this, your typical length scale would be the radius r. Then what would appear is the bending modulus b and the Young's modulus y, which is the in-plane Young's modulus. And you will get what is called a Foppel-von Karman number, which tells you how strongly nonlinear your problem is. Okay. And then there is a ratio of the temperature to the bending rigidity, which tells you the elastothermal number, which tells you how important are the effects of temperature. Now forget the G1 and calculate the spectrum for G0. Okay. You do it again in Fourier space, you get something like this. Okay. Now you can see for certain values of Q, which is the Fourier mode, your bottom can go to 0. What does it mean, the bottom going to 0? That means the spectrum actually blows up, right? That means there is a particular Q mode where all the energy goes and sits, it basically blows up. That is the signature of buckling. So I should have brought something where I could show buckling. So, but think of a football, okay? So think of this. So this is a simulation. So this is a shell and there is a pressure outside. If the pressure inside is slightly lower than the pressure outside, you can think that the shell is squeezed a bit. But if it is beyond the threshold, it collapses. Okay, like a football which collapses. And that's the buckling. And you see it selects a particular mode. So it is that mode that you will pick up from here. It comes within the linear theory itself. So the blow up of the linear theory gives you the buckling. Okay. Now you want to go beyond the linear theory. How will you do that? So going beyond the linear theory is using a field theoretic method is what I call is a physicist superpower. See, physicists mostly use techniques from math, right? Except this kind of field theoretic tricks which mathematicians are not so familiar with. So this is really the superpower of theoretical physicists. Okay? Yes. Makes very nasty. Right? Fourier is so much better. The L+1 things become such a nasty expression. Anyhow, this is not my work.
Now, can I ask a quick question?
Absolutely.
This Fourier method, is it effective when it comes to nonlinear equation? What is the justification?
So if you Fourier transform, then your linear part is just going to be fq f minus q, right?
Yeah.
The nonlinear part, for example, f to the 4 is going to be fq1, fq2, fq3, fq4 multiplied by a delta function of q1, q2, q3, q4 and the integral, right? That is the vertex here. So if you look at A in the figure, you see q1, q2, q3, q4, 4 legs connected by this dashed line. That is the fourth order nonlinear term. And then you have a f cube term, which is the vertex in the B. Okay? Is that clear?
Yeah.
Okay. Yeah. So now you can think, see, there are many ways to do this. Yes. Sorry? They are Feynman diagrams. Yes. And now you can ask, what the hell is a Feynman diagram doing here? Right? But that's the theoretical physicist superpower, to use Feynman diagram to brush their teeth. Whatever you do, you use Feynman diagram. So anyhow, jokes apart. So what you are doing is, then you are basically saying, look, if I look at only this part, then there is some source which is giving me an effect. But then those sources coming in and coupling with the vertex, and then every vertex is changing. And these are the leading order contribution from the vertices. But if you do this business, what you will realize is that many of these integrals will actually blow up. Okay? Sometimes they will blow up at large q, sometimes they will blow up at small q. Blow up at large q is not a problem for us, because we are looking at a system which naturally has a large q cutoff. Right? Large q means small space. And if you go to small enough space, this theory is not valid. But the small q blow up at the problem. And this is the Wilson's genius of finding a way to go around that. So what Wilson says is, write your theory in Fourier space, and then you have a cutoff lambda. And you do these integrals over a small shell, okay, very small shell. And then you have some corrections to each of the terms that are appearing. And you observe them as if every coupling constant in your theory is changing. So what is a coupling constant? B is a coupling constant. Pressure is a coupling constant. Young's modulus is a coupling constant. So you integrate over a small shell and each of them changed. And in the field theory language, these are called beta equations, the equations that are written down here. Okay. So as you change scale, you get these equations of how each of these things are going to change. Okay. So you're basically saying, I take the nonlinearity, I solve the problem, but I don't care about every length scale. I only ask questions at large length scales. Then what happens is, the coefficients appearing in the linear problem change depending on the length scale you are looking at. So that's the renormalization. Okay. And these are called the RG equations. Once you do that, you get buckling. Okay. So once you go do that, what you see is that the thermal buckling, the buckling that was there in the linear theory changes depending on your temperature, and sometimes changes quite drastically. So over here, I am plotting, for example, the critical buckling pressure, and you see for small temperature, it is 1. And that is basically means that what you would get from theory of shells in mechanics. But as you are doing statistical mechanics, you see the buckling pressure actually decreases and even can become 0. So a shell which is that way cannot sustain itself, it will collapse by itself. Okay. These are kind of fascinating theories, comes from Paulos and Nelson, and then this is a name I can't pronounce, and Nelson. So these are theories, these are very interesting papers with theory and simulations. But the reason I talk about this is that I want to talk about active shells. So this is the story of active shells that was invented, and I have forgotten to write the reference here, by a group of Jacques Pro at some time in the late 90s, in the last century. What he did was he took a bilipid layer and then put some proteins inside. And then when he shines light on it, the protein starts fluctuating. I mean, they start as ion pump. And then this shell is no longer a passive thermal shell. It becomes an active shell. And the question we are asking is, what about the buckling properties of these active shells? Okay, but we are doing simulations. So you now need to ask, what is the right model for activity? How do I include activity in membranes? Okay, so the Jacques Pro and company already did that. What they did was they took the membrane model and added a noise to it. And that noise was actually an Ornstein-Uhlenbeck noise. So this is very much like the model that Bishwajit worked with. Okay, so the simplest model of activity is to add an active noise, which is Ornstein-Uhlenbeck. Okay, but we are using a trick which was invented by Chandan Dasgupta and Manoj Kumar a couple of years back, which is extremely cute. So if you remember your statistical physics, you remember that when you are doing a Monte Carlo simulation, what you do is you go to a point and you say, I move this point and I calculate the change in energy. If the change in energy is decreasing, then I accept the move. If it is not decreasing but increasing, I still can accept it with a probability of e to the power minus beta e. Okay, and if you do that, you will get the thermal equilibrium. And between that is the idea of detailed balance. Okay, but you can break the detailed balance by saying that I make a move, I get a change in energy, then I add a constant term to that change in energy, and then either accept or reject it. So that constant term could be both positive and negative. Okay, either way you will break detailed balance, and in both the cases you will not see equilibrium feature. You will see, okay, so this part of it has to do with the equilibrium Monte Carlo of spherical shells. We wrote a paper in, so this is called Journal of Open Science, Open Software or something like that. You can write your code and put it there, and this is now open source and anyone can use it. But I have no time to discuss this Monte Carlo, so I will skip over that. If anyone is curious, I can talk about it later. If you want to use the code, I am overjoyed. So what is happening, so what we are doing next is, you see, I told you that there is an amount by which I am breaking detailed balance. If this amount is positive, that means I get fluctuations which are more than equilibrium. If this amount is negative, I get fluctuations that are less than equilibrium, but both are non-equilibrium. Okay. So in the middle are the two cases which are thermal, which is activity equal to 0. Now in the right, I have added things which are more fluctuation, and I see buckling. Okay, so something that is not buckled in equilibrium, if I shine light on it, that is going to buckle. Okay, that's this thing. And something that is buckled, okay, if I can make it more calm, it is going to unbuckle. That is the main result of the simulations. Is that clear? Maybe I'm going too fast, but I really have no time. Okay. So from this, I can draw a phase diagram. I can plot pressure on one side and the elastothermal number on one side. And then I can draw what is the phase diagram between buckled and unbuckled for various activity. So this dark line, this black line is from the theory of Nelson. And this red dots are their simulations, okay? And this blue line is from our simulation for the thermal case. And you see it is not very different from their simulations. The agreement is quite reasonable. But the things change when you put in activity. It can either go more buckling or less buckling. And here is the same thing plotted in the pressure by the activity phase space. So all that is detailed. But the main idea of it is the following. Let's say you have a microfluidic device. And on its surface, you have made some small microscopic shells. Each of these shells have these active proteins in them. Then you choose one and shine light on it, and that buckles. And then you remove your light, that unbuckles. You put your light on another one, that buckles. And you remove it, and that unbuckles. If you choose your parameters properly, you can do that. And then you can play on these shells like a musical instrument. Okay? And then you can imagine each buckling or unbuckling is working like a bellow. So you can drive the fluid flow at very small scale in an extremely controlled way. Of course, all that is just based on simulation, but that is the main purpose of this paper. Okay? No. Crumpling is different. Crumpling is when your correlation function of the height has certain behavior. But the buckling is a global phenomena. Crumpling can happen in a plane membrane. Buckling typically doesn't happen in a plane membrane. Yes, but you have to look at different parameters. Buckling is really equivalent to this. Okay, so this is buckling of a filament, whereas I was talking about buckling of a shell. Okay. Anything else? Okay. So I am almost done. Okay, if you really want to be tortured. So the last part is the part where I am going to talk about how random activity can make my whole problem nonlinear. So I've already told you about elasticity, so I'm going to go fast on that. So you see, this is the equation of an elastic medium, where you say how velocity changes is the divergence of a stress. Okay, but now think of the elastic medium, and you think that some parts are active and some parts are not. Okay, and which part is active is random, and it is random and fluctuating. So when it is random, then you can model it by saying that I have some Young's modulus in the whole medium, and then there is a fluctuating part of the Young's modulus. So there are two noise in the system. One is the noise of the Young's modulus, and the other is the thermal noise. Okay, so you can do a theory of that. So this is the fluctuating part of the Young's modulus, and this is the correlation function of that. Okay, so if you do a theory of that, no, I'm assuming the Young's modulus has one part that is constant, other part that is fluctuating in space and time. And I'm assuming that random thing has a correlation function. Okay, so then I have this kind of a differential or partial differential equation. Okay, that partial differential equation can be turned into a path integral using something called Martin-Cgiarose method. Okay, this path integral is harmonic, but it depends on two noises. The other set of problems where we do this is Navier-Stokes. So you would say there is Navier-Stokes equation and there is a noise. And that partial differential equation is equivalent to a path integral. And that path integral, again, you can calculate with this method. Now you can then ask, why have we not solved turbulence? Because then the Feynman diagrams that you get, they have very nasty properties, including divergences at small q, which we don't know how to cure. That's a long discussion. No, I'm exactly talking about something that is composed of active matter and inactive matter. So one part is this, which is constant, and the another part is this, which is fluctuating. So I know, but then you are normally come back. Yes.
but you are assuming that one product is uniform, and then you are assuming that everything is also fluctuating.
No. That's a very good question. I am basically assuming that I have a uniform matter with some random things embedded inside. And I'm looking at a scale that is large enough that includes many random fluctuating things and a homogeneous background.
So you are saying that the active as well as the reactive matter are fluctuating randomly.
The inactive is constant.
Then why are you not just simply modeling the fluctuations as something that is specially inhomogeneous?
That's exactly what I'm doing. So the fluctuations are something that but you see that spatial inhomogeneous thing has to be random. So that I'm basically saying the correlation function of that random quantity is this. Okay, so this kind of curly y is the fluctuating part, and this is the constant part. The Young's modulus is some constant plus some part that is changing. And it is this random part that has some correlation function. Okay. No, that depends on how you cut off your correlation function. You can say the correlation function falls off quite fast. That means this randomness is independent of that randomness and so on. So that you can all build into this function. Absolutely, yes. I am building a concept of coherence length in the randomness. Absolutely. So then I get a path integral which is harmonic but depends on two noises. If I average over one of them, it becomes biquadratic. And then I can do a perturbative RG. So when I came to this, I got tired of writing a slide, so I gave up. But if I do the perturbative RG, the Feynman diagrams are essentially those of what is called the 5-4 model. So someone has already calculated that for me. So with that, I can calculate the renormalized elastic coefficient. But the beauty of this is, what is happening is, the activity is making your problem nonlinear. So there are various people I need to acknowledge now. So these are the collaborators: Srikant, Vipin, my student, Vikash, who is a postdoc at NORDITA, Supriya, who is at Stockholm University, Shubhro, Abhijit, Vishwajit, and Ayan, somewhere there. Tonmoy, Tithi, Orikta, and Bidisha. Bidisha could not come because Mrs. Banerjee didn't allow her to, but rest of you are here. And I am really grateful to Ceci, for which it was possible to come and visit this time. And I'm also really grateful that they allowed me to do this kind of things which are clearly not astrophysics. The visit is sponsored by Spark, and these are the agency that feeds me meatballs. So to summarize, we show how to calculate entropy generation rate at very small scale over very short times. We can also show that buckling of elastic shells can be controlled by activity. And there are many surprises in active solid. For example, one is that the activity can generate nonlinearity, but there is also this question of generalizing Kubo formula for the active elastic solids. Thank you very much for your patience. You have been extremely kind.
Yeah, so it has been a pretty long
Oh.
Can I ask a question? Am I audible?
Absolutely. Yes, please.
Hello. Yeah. So when you are saying active matter, it's not just time dependence, is it? It's like the matter has free will, and that is introduced through the randomness, is it?
No, free will is more difficult.
I see.
Right.
So then, why the randomness? I mean, in a sense,
So-
active is like saying that it's time dependent, but on top of it, randomness is from the free will, or no, or lack of knowledge.
Lack of knowledge is one thing. So it is, see, there is, so as I mentioned before, I was going to say what I call the Anna Karenina theorem. So there is a part in Anna Karenina in the beginning which says that all happy families are alike, all unhappy families are unhappy in their own ways. So the generalization of that is all equilibrium systems are the same, all non-equilibrium systems are non-equilibrium in their own ways. So there is no one model of activity, there are many models of activity. But the simplest model that people use is to add what they call an active noise. and that is typically a time correlated noise. So far I have mostly stayed within that model, and that noise can be both in space and time. So for example, if you are thinking of a cytoskeleton, you don't know, for example, exactly which motor is working where and pulling whom and so on, right? So you will replace that by saying that the cytoskeleton is active, and in addition to the thermal noise, there is some active noise. Now, Tanmay will tell you that this model where you just add a noise can actually be wrong. This is something we are learning now, and Tanmay is very secretive about it, so he will not tell you about it now, but maybe next month.
Okay. And the other question followed up with this is that as you introduce time dependence and time dependent noise, can one not extend the Wilson RG in the frequency domain together with the momentum domain to write perturbative RG? And
this is actually in frequency domain, and see, all that I am doing in the time dependent thing is I am adding an Ornstein-Uhlenbeck process, right? That basically means if I just increase the size, the dimension of my system, I am back to a system with delta correlated noise. So this is really cheap game, just made to look fancy by sticks and stones. But
no, okay, so maybe I clarify. When we do the Wilson RG, we take the K and K plus delta K shells, and then we go ahead and do RG. Now, rather than just doing that, you add frequency to it and do omega and omega plus delta omega.
When you are doing
omega, yeah.
When you are doing
Please
Wilson RG for an equilibrium system, yes, you are right.
Yeah.
But when you are doing Wilson RG for a time-dependent system, then it is a large time, and also, I'm sorry, there is a large length and also large time. So yes, you are also going to small frequency.
But is the now the shelling is in the K omega space? Do you do the
Shelling
shelling
is
in
the
the
absolutely is in the K omega space, but you typically do it in 2 steps.
I see.
So there is a paper, this Martin CGR Rose is one. There is a paper by Foster, Nelson, Stephen, and this kind of goes back to mid-70s. This is done, I think the cleanest case is what is called the Kpz equation. I think there is book by Kardar, and the second volume he actually does these things.
I see. And then now the question for the first part of the talk. So when you define this rate of change of entropy, is it possible to define like entropy currents? I know people were interested in from the perspective of information flow, so can one define in a controlled way entropy current and then conservation laws and continuity equation, maybe source term? But and is it possible? Or
maybe you can, but we typically don't think of entropy as a conserved variable, right?
Surely, that's why I'm asking you this. But then it means it has a source or a sink, and then I can probably write in the sensible equation.
But there may also be boundary terms.
Yeah.
कल भी यही बना दूं, तुम कहो तो डेली बना दूंगी, हेल्दी भी। हाँ बना दो यार, बना दो।
I can't answer that question.
पता नहीं लगा पाओगे कि इसमें क्या क्या पड़ा है।
ये तो मैं पता नहीं लगा पा रहा हूँ, क्या डाला है तुम्हारे सीक्रेट इंग्रेडिएंट तो पता ही नहीं चल पाते यार।
This is strange. Anyway, thank you. I don't know what's going on in the background. So thank you.
I can honestly say I have no answer to that. Thank you for your time. Sure. দাঁড়াও, এটাকে অফ করি আগে।
Specific things which is it would have been nice to write it down somewhere or draw somewhere.
We don't keep a board here.
এটা কি তোলা যাবে? এটা তোলা যাবে? এখানে না, এখানে 5 মিনিটের একটা জিনিস করব।
Very nice.
Oh, thank you.
Yeah, it's
Thank you.
So one thing, so this the time.
Oh, thank you. Thank you.
I have a few questions on entropy production. I will ask you.
See, you are
I
the
have
the person I was most looking forward to talk to, because you are much better and an expert in this than me.
No, not in this area the way you were working.
No, but I am a novice.
I have small thing to talk about just linear response.
So this XT, sorry.
I just so the point is the following. Lot of what we do is we try to capture the free energy landscape as a function of some reaction point. It could be a multidimensional free energy landscape also. So let's say it's some kind of a landscape of this. Then we don't know. Usually what happens is we get caught into one of these energy minima and we take like really long something to get to, to get into equilibrium. So we think we don't know whether we are in equilibrium or not. Because in equilibrium, then these have to be both one sample in the right? That's usually not the case. So what we do is we trick around. We apply biases, right? Then we integrate out the right. Monte Carlo molecular dynamics, all the similar family. Now, so we are also into that game. Of course, defining the reaction coordinate or corrective variable itself is a second. Now, we are also building some methods to do this. How to